Hello, students. Welcome again for biology lecture. And last lecture, we were dealing with the molecular basis of inheritance. In that chapter, we have discussed so far that is replication of DNA. Replication of DNA, and this DNA replication takes place in the S phase of the cell cycle, and it happens only once in the life cycle. That replication of DNA differ in a prokaryotic as well as the eukaryotic type of the cell. Some differences are there, and in the prokaryotic, this replication occurs while the eukaryotic cell replication takes place at various places. That means the origin is of many points, but in a prokaryotic, it is only one. It is just like the theta shape, and hence the name is given theta. But replication in eukaryotic, it is at many sites or many points or the many origins. Let us discuss with the new topic in this chapter, and we are going to discuss that about this with the next point, that is how this replication, after the replication, the main factor or the main process takes place in the life, and that is protein synthesis. Okay, so in a protein synthesis, let us see what happens. So the protein synthesis, here the central dogma is there, central dogma. And let us start with the slideshow from the current slide. So this is the protein synthesis and central dogma. That means what is the basic need or the basic idea behind this protein synthesis, and how it process by the direction. What are the different steps? It is postulated first in a 1958 by FHC Crick, and he gives us the idea of the central dogma. So let us discuss about this. Here, the central dogma. Here, DNA, that the transcription takes place, and DNA synthesize mRNA. DNA, which is the master power, you can say, that it totally control all the types of the processes takes place in the cell, and which type of protein is to be synthesized. It is totally control, govern, and it is done by DNA. So from DNA, mRNA synthesis takes place. This process is called as the transcription. Now DNA is present inside the nucleus, and the synthesis also takes place inside the nucleus. This process is a transcription where mRNA synthesis occur. Now, what type of the protein is to be synthesized? It is totally controlled by DNA, and all the message or the coding is written on the DNA. According to that, that DNA act as a template, and this DNA which act as a template as per the written message, the mRNA synthesis takes place. Though this process is called as a transcription. After the mRNA synthesis, then the actually the protein synthesis process takes place. So this newly formed synthesized mRNA transferred from the nucleus through the nuclear pore to the cytoplasm. That is the site of protein synthesis. And this mRNA that goes or travel towards the cytoplasm of the cell, and then this, according to the written things in the codon from the mRNA, the translation takes place and polypeptide chain that is developed. So according to that, different amino acids are joined with one another to form the specific type of the protein. So this process is called as the central dogma. So the first step is transcription, DNA to mRNA. Message is given, so it is messenger RNA, and from messenger RNA, translation occur. So from the mRNA, the amino acids they link with one another. So that is a polypeptide chain of amino acid, and that synthesize into the protein. So proteins are synthesized according to the order of this DNA. Now, this concept, that is a central dogma, uh, it was uh, accepted as a unidirectional. That means from DNA to mRNA and from mRNA to polypeptide. But here, the new concept is there, which is a central dogma known as retroviruses or riboviruses, that is given by Timin in 1970, and second scientist, that is the Baltimore in 1970. Both these workers, they showed us that such a type of the transcription, translation, that is a synthesis of DNA to mRNA, can be have the reverse, that is from mRNA DNA may get synthesized. So in this way, this is the previously it was known that it is a unidirectional, but now it is not like that, but it is a concept given by these two scientists in 1970, and hence from mRNA again DNA can be synthesized. So next thing about this, let us discuss the uh, process of transcription, how it takes place in detail. So the RNA polymerase that act as a important enzyme, and this transcription process, how it takes place. So from the initiation, then elongation and termination. So this is the transcription, and DNA act as a template. So various enzymes act here. For example, RNA polymerase, and this process takes place in the nucleus, that is the nucleoid, you can say, and this transcription from DNA to RNA, it occurs in the cell during the G1 and G2 phase. Now, before that, in S phase, what we have discussed, that is the formation of DNA or the replication of DNA takes place only in the S phase of the cell. But these are the two phases, G1 and G2, that is the post mitotic and pre mitotic phases. In this period, in this condition of the cell cycle or cell span, this transcription takes place from DNA to RNA. So, this is the idea of this transcription where it takes place in the phase of the life cycle of cell. Then, now let us see in a detail about this. If the template strand, it has a 3-5 direction. As you know that there are two strands of the DNA, and these two strands, they are arranged in an anti-parallel manner. Means what? If one has the origin 3 to 5 direction, then other strand it must be 5-3 direction. So it is anti-parallel, as well as having the arrangement of these two strands is with opposite manner. So one is ascending, you can say, and second is the descending. So it is here the template strand, and then structural gene and the coding strand. See here what are the things which are important. So RNA polymerase that play an important role during this formation, that is transcription process. So this is a 5-3 direction. You can see here this is the 5-3 direction, and the another one, that is 3-5 direction. Oh, sorry, 5-3 direction. So in this way, these are the two strands, and this region it is a promoter, and here the structural gene, template strand, and coding strand. So structural gene, what message uh, is given to the synthesis of mRNA, that is given on the one strand, that is called as a template strand, and actually the coding strand, this is the real message that is given to the mRNA. Now, mRNA, one more thing that we have to understand, or we must keep in mind, that the first thing is that this RNA synthesis takes place from the DNA, but there is a change, or there is a difference between DNA and RNA, is that major thing, that is about the thymine. So in DNA, thymine is present, but it is replaced when RNA is synthesized, it is replaced by uracil. So we must keep this in mind that when the RNA are synthesized, but the changes are there where thymine is replaced by the uracil. Next thing that you can see, or these are the most, these are more convenient things that we can get the idea of this. The first one, that is the mRNA at this side, left side, and in between these two, that is the coding strand and mRNA, the template strand is there. So the direction of this template strand is 5'3". Okay, so we can see here the cursor, and 5'3" direction where this nitrogen bases are arranged. That is the purines and pyrimidines. They are like this. So this is a C A T G T A C. So all the direction is uh, very accurate, or it you can say it is a specific, and it is the 5'3". If it is changed, the meaning of amino acid and protein is also changed. So this template strand act as a template or the mold, which type of the codes or the message is to be given for the synthesis of mRNA. So according to the template region, complementary strands, or you can say complementary nitrogen bases, they are synthesized on the mRNA. So here you can see here uh, this in front of this 5'3", where here is a C, but is complementary thing is that G is there. Then A, that having U. Now see here the difference. Actually, in DNA, adenine that is always pairs with the thymine. But what I said before, that the instead of the thymine, uracil is replaced, and so that all the complementary nitrogen bases are uh, totally transcribed to the mRNA. If you see this mRNA, messenger RNA with this coding strand of DNA, both are exactly same. So the direction is also same. See here, it is a 5'3" direction, and at the base G. Same thing here at mRNA G. Then here it is U. Then here it is a T. So in this way, they are exactly similar. So what I said, that T, that is replaced by U, and hence thymine is not there, but the uracil is there, and remaining all purines and pyrimidines, they are similar one. So in this way, this is a central dogma. You can say that the transcription process takes place, and basic things we must know that is given in this chart, or you can see in your book also. Now this is all about transcription process. Next, here Uh, there are three types of the polymerase. These are the three polymerase: RNA polymerase first, second, and RNA polymerase third. Now here they are mainly functions for the synthesis of RNA. So polymerase first, that involve in the RNA, then polymerase second, that involve in the next one, that is having such a type of arrangement where rRNA, and the third polymerase that is concerned with the tRNA synthesis. So in this way, such a type of the polymerase types are involved in the synthesis of different types of the RNA. Now next, let us see about this transcription unit and the gene. Now this DNA sequence coding for mRNA or tRNA or RNA, that is called as the gene. So this is the sequence of DNA, and these are the codons. Now the segment is there on the DNA, which is called as the cistron. So this is the cistron is the segment, and it is the DNA coding for polypeptide. While when it is a single structure of the gene, then it is called as the monocistronic. While when it is a long segment of DNA with the various structural genes, then it is called as the polycistronic. As the name indicates, has the same meaning. Monocistronic means the single structural gene, while the polycistronic means they have many or the various or they have the long segment of DNA. So these are the various structures of the gene polycystron. Then there are two parts on the mRNA. You can see here introns and the exons. Introns, they are non-coding sequence in structural gene, and actually these introns, they do not take part in the inheritance or the transfer of the characters from one to next generation. But the exons, they express the sequence, and that is important. If you see in the prokaryotic type, introns are not there, but only exons are there, and hence during this process, that is the transcription process, there are no further processes are present in the prokaryotic type when the mRNA synthesis takes place. But when it is a eukaryotic type, then these introns are cut, they are removed. You can see splicing is there of the introns, and only exons, they get joined with one another. So in this way, this is all about the transcription unit and gene, which the necessary things we must know, those are given here. Now next one, that is the process, actually what this process takes place of the mRNA synthesis. So eukaryotes we are dealing here, that is about the eukaryote cell. See here, the eukaryotic DNA, again having this, just like the central dogma, this is the primary transcription where this eukaryotes DNA totally converted into hnRNA, which are the hnRNA means having this process where they represent the RNA, and after this, that is the heterogeneous, you can say heterogeneous nucleotide RNA, and this is a splicing, capping, and tealing. These are the three processes for the synthesis of mRNA. So this is the three processes which require for the processes of mRNA. So here, this um, formation of mRNA, that requires, that means when the RNA is synthesized, it is not utilized immediately, but it has to go some processes, and those are known as the splicing, capping, and tailing. So what are these different processes? Let us see here. So this is the DNA, double stranded structure DNA, and this portion, what I am showing here, this portion uh, where these codons are given, according to that mRNA synthesis takes place, and from this total mRNA synthesis takes place, and you can see here there are two regions, some dark and the faint. So dark is called as the intron, and faint is called as the exon. So as per the knowledge that introns, those are totally removed. Introns are totally removed, and only exon get joined with one another to form the segment. See here, this process of capping that starts, and sorry, huh, this introns, those are removed. So this is a newly formed RNA, mRNA, and the introns are removed. And at both the ends, there is the addition of different things. That is the at the one end, it is a polyadenylation X plus. At the three end, polyadenylation, and second thing is, that is the towards the tail end, you can see here polyadenylation takes place, while at the phi end there is a uh, GPPP, and the addition of this GPP takes place, which is known as the capping. So capping and tailing takes place, and third thing is that the splicing, that is the introns are removed. Now you can see here, this is the segment of mRNA. Now this segment, that is ready for the further processes. So which is the further process? That this mRNA transferred from the nucleoplasm to the cytoplasm, that is the site of protein synthesis. So this 5-3 mRNA, now it is ready for to give the message which type of the protein is to be synthesized. So in this way, this process of mRNA takes place, and that is complete. in the three processes were included, that is having this heterogeneous RNA, that is HN RNA, and then it undergo splicing, capping, and tailing. So splicing, that is the cutting of intron, which is not utilized. That means the exons which carry genetic information but not intron. And the second is the capping, that's the uh, towards the front end, you can say cap like region. There is the addition of GPP, and towards the tail end, there is the addition of polyadenylation. So in this way, 5' messenger RNA that is now transferred from the nucleoplasm to the site of protein synthesis, that is the cytoplasm. So in this way, this is the all about in short. Now next thing is that before to go in all processes of protein synthesis, we must know how this genetic code that is totally included, or how they have the combinations, permutation and combination. You can say that we know that there are uh, four nitrogen bases, two purines and two pyrimidines. The uracil, cytosine, adenine, and guanine. So these are the four. And how they are arranged? Let us see here. Here it is written first letter. For example, the U first, C, A, G. So in the code for codon formation, this is to the first site. Then here it is written second letter, the same uh, nitrogen bases, purines and pyrimidine, UCAG. And here at the third, you can see UCAG. So at these three letters, they are arranged with permutation and combination and repetition. As you know that there are 20 amino acids and having the four uh, nitrogen bases, how they are arranged with the permutation and combination. And they are having the three triplet forms. So in this way, you can see here this U at the first letter, then this is second U, and again the third one, that is U. So in this way, this is a U. So this represent a particular type of the amino acid. Then second UC. That means the first two are same one, U and U. This is second letter, but the here third letter is instead of the U, C is there, and so on. The third letter here it is changed. It is a A. So in this way, this is the permutation and combination, and it should be triplet form, and all these four uh, nitrogen bases, that is the purine and pyrimidine, they are arranged in this manner. If you see this total, there are four, and the arrangement is like this, so that you will get about the 64 combinations, 64, and this are they represent a particular type of the amino acids, and these amino acids are arranged as per the requirement. So as per the specification of the protein synthesis, all they are arranged in this manner. We can arrange this, which is called as the dictionary of genetic code. So this genetic code, with this permutation and combination, they represent a specific type of the amino acids. So this is all about genetic code and the arrangement of this genetic code that is given in this table. So before starting of all this protein synthesis process, we must know about this. Uh, Now we have to stop here because of the time factor, and we will discuss this to the next lecture. We are going to see or discuss about the characteristics of genetic code in the next lecture. So let us stop here, and we will meet again to the next lecture, continuing with the protein synthesis process. Okay.